lab here at Rockefeller, and today what I'm going to tell you about is developmental cell death and CLEs. So the two main things that I want you to come away with knowing after my talk is what CLEs is, why we use it in the lab, and why cell death is important during development, and how CLEs can be useful in studying cell death. So this is a picture, actually a very beautiful picture of the worm, Caenorhabditis elegans, and I'll just refer to it in shorthand as C elegans for the rest of the talk. So to switch to just thinking about cell death, my talk moves forward. Not a good day for a PowerPoint. Okay, so cell death is important for development, for normal development, and many of you may not necessarily be thinking about cell death, which is quite a morbid topic, but for an animal to develop normally, we really need the proper balance of the right number of cells dividing to make new cells, balanced by the right number of cells that are dying. So in fact, even in animals where cell death cannot occur, they can't reach development. So it's that essential for normal development for the majority of animals, including us. So cell death during development, so I'm talking about when an organism starts as an embryo, as a single-celled embryo, and divides to become an adult. Cell death is used to sculpt structures. So for example, we're all born with webbing between our digits, and cell death actually then carves out our individual fingers during development. Also, cell death is used, again, similarly for sculpting. So, for example, if you have a mass of cells, the cells in the inside of this ball will die to create a cavity. So, for example, during mouse embryo development, you have a cavity formed to allow the embryo to develop. Cell death is also used for deleting whole structures. So, for example, vestigial structures that still are created as the organism is developed, but is no longer needed to be built. So, the classic example would be a tadpole. The tadpole starts off with a tail and swims, and then as it develops to become a frog, cell death actually deletes the entire tail of the tadpole. Similarly, in us, cell death is used to create sexual dimorphism. So what I mean by that is as we're developing, we're very young embryos, we can't tell the difference between males and females. So we have precursor structures for both male and female organs, and one will die in the female, and then that'll generate the male, and vice versa. So for example, this precursor structure, which will go on to become the oviduct in females, dies in males. And then also similarly, a precursor structure that goes on to become the vas deferens and other male sex structures die in females. Another area where cell death is used during development is to adjust cell numbers. So just the way we happen to be developed is we have an excess number of cells that are produced, and then cell death actually scales back the number of cells that we have. So for example, in this case, it's illustrating that you have an excess number of cells. So for example, actually 90% of cells born in developing brain die. And so basically that allows for the proper connections to be made. So in the nervous system, you have your nerve cells, and then those are wrapped around the other cells. So you just have a ton of both of these cells made, and then cell death creates the proper matching of one neuron to one sheathy cell. Finally, also, cell death is used for more quality control mechanisms, and to maintain homeostasis. So if you have DNA damage from the sun, then that'll signal the cell to die, and that helps us not get cancer in those cases. Also, cell death is widely used in the immune system. So you have developing lymphocytes die. normally should not react to your own, should not cause an immune reaction to your own cells. And so if the immune cells are too reactive, then they'll get a signal and those cells will die. So these are just kind of groupings of sculpting, deleting structures, adjusting cell number, and eliminating dangerous or injured cells are ways that cell death is used during development. So now, how do we study cell death? One way to do it is to use a model organism, such as C. elegans. And so we use, I don't know if you're familiar with model organism, but essentially we just use another organism so that we can actually do the experiments in them. So for example, we can do many experiments in humans, and so as a proxy for that, we make mutants in worms as opposed to making mutants in humans. And so what I've listed here are some features that make C. elegans an excellent organism to study in the lab. So quite, a lot of these come down to just practicality issues. So for example, C. elegans, it's actually when we first studied. So C. elegans, it's a nematode, so it's a small microscopic worm that normally lives in the soil, and you can easily cultivate them in the lab. So you just have a small petri dish, and thousands of worms can grow on that. They have a very short generation time, so from embryo to adult, it takes 3 days to develop. It's very quick, so you can do a lot of experiments. They're small, so they're only 1 millimeter, so they're actually the size of a printed period. So 12.5 period, that's the size of the worm. So when we do all our studies in second microscope. They're a fairly simple organism. The adult only has 959 cells. They are quite genetically tractable, so this was actually the first multicellular organism to have its entire genome sequenced, and so we know all the genes that animal has. So it's very easy. Once we find our favorite gene, we can study that and make new mutants all the time. And another practical feature that C. elegans has is that they're transparent. So they're very easy to image. And so I should also just say, the worm that also makes it convenient to study is that there's 2 sex forms. There's hermaphrodite, so that means that they both have the male and female sex organs, so they can self-fertilize, and then males exist. And so that allows for easy genetic manipulation. So just getting to the fact that the parent has made it really easy to just visualize the cells. So here, what I'm going to show you is a time-lapse movie starting at the 2-cell embryo all the way through the entire embryonic development just before it hatches. So as you can see, this outline is the entire embryo. So there's a very tough cuticle that keeps the embryo enclosed from the environment. And then this is one cell, and this is another cell. And so this is just a time-lapse movie. You can see the cells dividing. Spinning around, flipping, and it's just moving around. So there you go. So that's going from the 2-cell stage all the way to completing embryogenesis to the first larval stage. So at this point, the animal will essentially eat its way out. Any questions?
How much time?
Okay. So that is about 14 hours. So part of this happens inside the animal, then the embryos are hatched, laid, and then development occurs ex vivo. And so, what a couple of scientists by the name of John Sulson and John White did is that they actually sat and watched going from the 2-cell stage, they watched every single cell division and every single cell death. So they have an entire map of how an adult organism is made. And they actually won the Nobel Prize for doing that. And so this, by just watching those movies over and over again and watching which cell divides came there, they made a cell lineage map. And so let me play with that. So this is this little branch over here. This is the first zygote, so that's starting as fertilized egg. You have a cell division, so you have a branch, and then you have 2 cells. And then this cell then divides into another cell, and then this cell divides again, and then you can follow the branches, and each of these branch point represents a cell division. And this is as time goes on. So here you have a complete map of how an adult organism is made. And this is extremely powerful because the worm has a fixed cell lineage, so they were able to do it. So it really is a very good for animals. You can look at tons of embryos, and they all divide in this pattern. And so just kind of, just make sure you follow this to zoom in this section. When you're looking at the cell lineage, the branch point is cell division, as I said, and so here the cell divides again, and this line ending means that it's a differentiated cell. So that's the terminal state that the cell will end up in. So differentiated state means that it has certain identity. So for example, this could be a muscle cell or an intestinal cell. And so its sister cell continues to divide, and one of these cells dies. And then you just continue along this pattern for the entire lifespan of the organism until it reaches adulthood. And so, as I said, alluded to before, this is really powerful, particularly for studying cell death, because we have an exact map, a roadmap of which cells die, when they die in development, and even the location, which position the cells are in. So we can start to think about how we could study cell death. So how can we study cell death in Elegans? So I've told you that there's a lot of great features about the worm that makes it easy to study in the lab. And what some researchers did, because they were interested in how cells are able to die, the genes, the genetic pathway for how cells die is not known. And so they used the power that C. elegans offered to identify the genes required for cells to die. And so simply what they did was they took animals, so here this is a wild type animal, so a normal animal, and the cells died. And then they looked for mutants, mutant animals in which cells don't die. So here you can see these lines which are what dead cells look like, and here you don't see any dead cells. And so by doing studies like this, looking for mutants for a given phenotype that you want, so the phenotype would be that cell death cannot occur, they were able to identify that the gene required for the cells to die is a gene called SED3, which is cell death defect number 3. And so this was very exciting, actually. In fact, won some of the Nobel Prize for identifying